So now we're going to see what is the chemical composition, structure, and selective permeability of cellular membranes. So cellular membranes are made out of phospholipid bilayer, and they will be having some proteins attached to them that can either go across these phospholipids or they just stay in one end or the other. If it stays in one end or the other, it will be called the peripheral proteins. If it goes all across the phospholipid bilayer, it will be called the transmembrane, trans-across membrane. And these lipids in the plasma membrane, they are not static. They're constantly rotating and moving. So this is what we call the fluid mosaic model, along with the movement of the proteins as well. So one of the main functions of these lipids and the proteins within the lipid is to get signals from the environment and to attach to other cells, but as well they create channels, these proteins, that allow the cells to be permeable to different molecules. Like we have channels for sodium, we have channels for potassium, etc. So this is what we call selectively permeable membrane, and not all the molecules can come in. They have to have the right charge, the right size. Okay, so they have then some of the ones that they have large size, they cannot pass through these small channels. So this restricts the movement and maintains the internal environment. So here is a magnification of the plasma membrane. So what you can see is this phospholipid bilayer. So this is the extracellular, so if this is the outside of the cell, this will be the extracellular leaflet, while this one will be the intracellular leaflet. So the fatty acids from these phospholipids faces medially, while the phosphate heads, which are these, faces externally in the environment and then internally inside the cells. Why? Because these ones, they have a charge that allows them to interact with water. And then here we have some examples of proteins. Another name of a transmembrane protein can be what we call integral. So this one goes across the two leaflets, the outer and the inner leaflet of the phospholipid bilayer. And some of them, they might have carbohydrate chains attached to them. If these proteins have a carbohydrate chain attached to them, they will be called glycoprotein, because glyco, it's kind of another name that we give to the carbohydrates. And then some of these integral proteins, they might not have these carbohydrates, but they can have attached to them some peripheral proteins, either out or in. And then some of these proteins will be anchored into the cell by part of the cytoskeleton that forms like a net, just right under the inner membrane of the leaflets. So then, because of this, many cells can attach to surfaces and they can communicate with other cells because these ones act as receptors. It's like having a doorbell in your front door. Okay, so it's like the receptor. Someone presses there and then it tells you that someone is outside there. Okay. So it receives a signal. So what are the functions of the plasma membrane proteins? The structure of proteins provide support and shape. Receptor proteins detect signals, and channel proteins allow the passage of molecules. Okay, someone ring your doorbell, you go and answer door, and then if it's a family member or friend, you let them in. That will be the channel. So now let's look at the different channels and carriers. So channel proteins have a pore, like a little opening, then that allows the crossing of molecules. So the crossing of the molecules can be going in, like this arrow is pointing, going into the cell. So everything from now on where you see this diagram, if it's kind of orange or yellow color, this is the cytosol, the inside of the cell. And here, if you see this ECF, means extracellular fluid, meaning that this space represents the outside of the cells. So this protein, this channel protein then allows the passage of this molecule in, but also can allow the molecule to go out. So in this case, you have molecules coming in and out, and then you can have carriers. So in carriers, you have actually a physical connection of the molecule with the channel. In this case, the movement is free, but in the carriers, the molecule has to bind into a gap inside the channel, and then that will open this gate or this part of the channel inside the cells, while this one closes. So it will be doing this. For receptors and enzymes. So receptors, they can bind a signaling molecule like a ligand, and then they can open in response to that ligand. So here we have this little gap in this channel that allows this molecule to bind, and after this molecule binds, this gate opens so that whatever molecule need to cross through here. It's like having a key to open a door. And then enzymes will be membrane proteins that catalyzes a chemical reaction. So sometimes we can have membrane-bound enzymes, like adenylate cyclase, the enzyme that can convert ATP into cyclic AMP. So in this case, you will have an enzyme that has internally these gaps, which are what we call the active sites, and then these substrates bind into these gaps, and then you end up with a product. So you catalyze a reaction. So other proteins function to support, like in this case, can be extracellular or intracellular proteins by binding these channels or these peripheral proteins, and then you will have a stronger location of these molecules, like these proteins, without moving so much around. Other ones, they can link adjacent cells. So in this case, two cells are nearby, and they form a junction, a connection. So we have what we call then linker proteins, because they bring the cells of this tissue together. Another name will be cell adhesion molecules. It's like a glue. So now let's look at the composition of intracellular and extracellular fluids. So the intracellular fluid, it is the fluid that is found inside the cells, and it has within it water, of course, and it has solutes. And many of these solutes will have a charge. So depending on the cell, you will have certain concentration of ions like calcium, magnesium, etc. And all the cells have more or less the same type of ions. So they can have calcium, they can have sodium, potassium, and the concentration of potassium in excitable cells is more increased inside the node. Potassium, it's a positive charge ion, so it's a cation. And then we have sodium as well, which is a cation that is also found inside the cells, just like as it's found outside of the cells. So if you have the same types of molecules in and out of the cells, the only thing that changes is how much concentration you have. So potassium is more abundant inside the cells than sodium. And outside of the cells, sodium is more abundant than in. And same for chloride. Chloride is more abundant outside the cells than inside the cells. Chloride, it's an anion, it has a negative charge. So let's say that this room is a cell, and then we have here students. So we basically will become these ions, and then outside we also have students maybe less abundant than you. So I can have, for instance, you are AMP1 students. So maybe there will be one or two that are coming in, but then the concentration right now is higher number of AMP1 students than outside. So another thing is that we have to maintain these differences between the concentrations so that your cells can be acting and functioning normally. So maintaining homeostasis. Now let's look at what makes these molecules to come in and out of them across the membranes. So several factors influence the movement of molecules across the membrane, including the size. The smaller molecules cross much easier. They have a greater amount of movement, kinetic energy, than the large molecules. The charge of the molecules. Well, nonpolar cross freely, and some of the ones that are polar, that they have a charge, they have to cross in a specific channel that it doesn't have the same charge. Like for instance, for potassium. So potassium is positively charged. So if you try to move a potassium molecule across a channel that has positive charges, it's not going to happen. It's not, because of the law of the opposite attracts. Positive with positive will repel. So then the channel has to have proteins that have negative charges so that this molecule can cross. And nonpolar molecules like CO2, oxygen, can diffuse the membranes really quick without a channel because they don't have a charge. Another thing that you need to have is a difference in concentration. So basically, that's a gradient, what we call a gradient. So molecules tend to move from higher concentration to lower concentration. And once you reach an equilibrium, the molecules won't move anymore. However, we have some pumps that helps to maintain the different concentrations without making it to reach an equilibrium. So the movement from high to low concentration is defined as diffusion and passive transport. Now, transport proteins, which are carriers and channels, they assist the molecules to transfer, but they will be saturated. So it's not like a conveyor belt that is constantly circling around, and then maybe if you put it next to a table, whatever you put in that conveyor belt is going to be going into that table. But if you want someone to carry objects and the person needs to perhaps load a pickup truck, and they had a lot of boxes there, the person, it has only 2 arms, so the person will be doing this. So it will be saturated. It won't be able to do all the transfer of all the boxes at once, unless, of course, he has, I don't remember the forklift. That's the only way, right? But then you can saturate your transporters. And then you have to have energy. Some of these pumps that helps to maintain the difference in concentration in the molecules, they require ATP. And these are called ATPase enzymes or channels, pumps. So if you don't have energy, you don't transport the molecules. And then membrane composition. Lipid and protein makeup affects the permeability. So if you have only lipids, but very little channels, you cannot transport polar molecules. And the more channels that you put, the more molecules are charged that you can transport. So for the electrochemical gradients, so it's not only concentration, but also the charge. As the molecules are moving in and out of the cells, the charge inside the cells changes. If more positive molecules move out, the cell becomes more negative, which at the same time as it's becoming more negative, it will tend to pull back the positive charges, as you will see in the action potential. So, of course, if you want to have movement of students in a classroom, you have to have a period in which, of course, there will be a space for the students to exit and then the next to come in. If only students come in, and then professors start teaching A&P, and the other one chemistry, physics, and all the 3 professors at the same time, and all the room is full, it doesn't make sense, right? So you have to have that exchange with the electrochemical gradient. So then how the passive transport processes work. So diffusion, again, is the movement of molecules across a semi-permeable membrane from an area of high concentration to low concentration. So molecules move from more to less. The other day we did this exercise, or why I asked you the question, like if that set of tables that it's in the middle, or there in front of me, were the plasma membrane. And then in here we have these channels. And then in here we are 3. And over there you are 10. And over there you are 11. What will be the normal flow from 11 to 3 or from 3 to 11? From more to less or from less to more? From high concentration of people to low concentration, from more concentration, from less concentration to low? So, to reach an equilibrium, 11 from this area where you have 11 persons, some people have to move in here, or some people from here to here. And then you reach equilibrium. From here to here, not here to here, right? And then how many people would have to move from here to here to reach equilibrium? So that we can have equal numbers. It's 14 in total, right? So 7 and 7. So we have to move 4. Right? So 4 people will move here. And then you end up with 7 here and 7 here. And that's how it works. So that's diffusion. And then again, in simple diffusion, you don't need a channel. So basically, non-polar solutes will pass through the plasma membrane. Oxygen and CO2 are gases that they don't have a specific channel to move across. They just go through the plasma membrane. And then facilitated diffusion, it is when you have molecules, again, that are polar charged, and then they move via carrier or a channel protein. Another type of movement is osmosis or osmosis. It is the movement of water across a membrane from higher concentration of water to lower concentration of water, or towards a higher concentration of solutes. So now let's talk about plasma membrane and osmosis. So if you have here the plasma membrane, and this is the cytosol, and this is the extracellular fluid, ECF. Let's say that someone ingests a food that is high in sodium, like pizza. So then your sodium levels, you have water here in your extracellular fluid. So you have water plus sodium. And then say that your concentration of sodium, this is how you write it down, goes high. after you eat this pizza. So you end up with a bunch of molecules of sodium in here. And then here in your cytosol, you also have water and sodium, but the concentration of sodium inside the cells is very little. Look at the size of the brackets. So you have only this amount of sodium. So you won't move the solutes, you won't move the sodium. You will move water in osmosis. So in this case, water is going to go from the cytosol into your extracellular space to dilute this amount of sodium that you have. And as you do that, as you move water, so water will move in order to dilute. So you will increase the volume of water in the extracellular space, and you reduce it in your cells, in your cytosol. So that process is called dehydration. So the cells will shrink. And then your brain, it will send a signal that tells you you need to drink water, so that you can compensate by losing water from the cytosol, and then you move back water, and then you don't feel thirsty anymore. So here we have an example of simple diffusion. This is a beaker with water. You have a plastic divider, and then in this side of the beaker, you have high concentration of glucose, 50%. On the other side of the beaker, you have 5% concentration of glucose. So it's a solution where you have glucose. And then if you remove the divider, eventually your solution will have equal number of glucose molecules across the water. So what will be the total concentration of glucose in that case? Left side, towards your left you have 50%, and on the right you have 5%. You remove the divider, the molecules will move from more to less until you reach equilibrium. So 4 people from there will move to here. We will end up with 7 and 7. What will be the total number of persons? 14. So what will be the concentration of glucose there? Say it loud. I can read lips, but
27.5%. If that's a percentage, can't work.
you have to add them, right? 55%. We don't change the number. You can move here, and then the total concentration will be 7 plus 7, 14. Okay, so now what's the difference between simple diffusion and facilitated diffusion? Well, simple diffusion is a passive process in which the nonpolar molecules move across the membrane without energy, and the movement is small. In facilitated diffusion, you have movement of larger molecules or polar by these channel proteins or this carrier, and no energy is required. So they have similarities, no energy required for any of them, but in one you use a carrier and the other one you just use a carrier. Then polar versus non-polar. Charged molecules, they need a channel. And then non-charged or non-polar, they don't require a channel. So now let's look at this image of simple and facilitated diffusion. So here we have our plasma membrane with a phospholipid bilayer. Again, this is the extracellular fluid, this is the cytosol, outside the cells, inside the cells, and then non-polar molecules diffuse freely because they don't have a charge. They are basically hydrophobic. And then you can have movement in and out. Like CO2 exits our cells, oxygen comes into our cells. And then for facilitated diffusion, we have ions crossing. And these ions will be specific. Let's say this one is a sodium channel, so only sodium comes in. And then this is the potassium channel, so potassium goes out. And then we have this pump that helps to maintain the equilibrium. Now for carriers, basically the facilitated diffusion can basically bring molecules that are polar or ionic compounds. And again, you can basically bring them in and out as long as these carriers have that particular size and shape to bring the molecules in and out. Our fingers are longer than our toes for a reason. I can carry with my toes one of these. If I'm lucky, I can grab it with my tiny toes, and then I can carry them, but my fingers are longer. So then these openings in the channels, they carry specific molecules. Like glucose, for instance, we have glucose carriers. So now those were modes of transport that they don't require energy. Now let's look at the ones that require energy, which we call active transport. So we have primary and secondary. In primary transport, you have ATP that directs the power of the pump so that you can move the molecules. So for instance, we have the sodium potassium ATPase that carries sodium and potassium. And then in secondary transport, you use energy stored in ion gradients to transport other molecules. So we have, for instance, sodium glucose transporter. And then you can transport sodium and glucose at the same time. Some of these electrolyte solutions that are used to hydrate someone who who is dehydrated, they have glucose in it, and they have sodium. And it is not to make the person more thirsty. It is because maybe that person is also losing energy, and then they have basically not only bringing sodium into the cells, which pull water into the cells, but also the cells will have energy, and then the transport can be more active because you're bringing glucose, which is used to make ATP. Like sometimes you can put in a slice of an apple, little lime juice, and then little salt. Tastes so good. You have the hydrogen ions from the lime juice, and then sodium, which our body typically craves for sodium sometimes, and then you have glucose from the banana. From the apple. Okay, so now let's look at how the primary transport and secondary active transport differ. So facilitated diffusion is passive, no energy is needed, and it is down the concentration gradient. Down the concentration gradient basically means from more to less. And then primary active transport uses ATP, and then you go against the concentration gradient. Now, we as three wanted to go into that side. And then you basically form a barrier, and then we start doing this, pushing against you. That's against the concentration gradient. And someone's going to take the elevator is full, person still needs to go to his appointment on time. What does the person has to do? Energy and then into there. Okay, so that's against the concentration gradient, from less to more. And then secondary active transport, again, you use these ion gradients to move molecules. And this is what we can call symport or antiport transport. In symport transport, you carry two molecules at the same time toward the same side. And then antiports basically exchange. So one molecule goes out, the other one goes in. So definition of osmosis: movement of water across a selectively permeable membrane towards higher solute concentration. So this one is different from diffusion because it involves only water, while in diffusion you involve basically solid molecules or solutes. And this movement of water is driven by solute concentration in and out of the cells. There are people who follow a diet that is not so much rich in sodium, and these people, they drink the regular amount of water that we all need to drink, but they don't feel as thirsty as someone who has a high sodium diet. And one way that I always use like an analogy for telling you about this high solute concentration, which is basically having a hypertonic solution, it is like, let's say that you're preparing a hot tea or a coffee in the morning if you like coffee, and then you are kind of half awake, and then you put the sugar in the cup before pouring the coffee. And then after you pour the coffee, you don't remember if you added sugar or not, and you add it again, the sugar into it. And then you mix it and you taste it. how does that coffee is going to taste to your like? If you have added more than what you sugar than what you usually add, how it's going to taste? Very sweet. So that solution is going to be hypertonic. Going to have a high concentration of sugar. So if you want to make it drinkable to your taste, what do you need to do? Add more coffee. That will dilute. So that's what we do with osmosis. We eat a lot of sugar, we eat a lot of salt, and then our body tells, drink water, you are so sugary, you are so salty. Unless you're rich, you will throw away that cup of coffee and you serve another one because coffee prices are going up. Okay? So I will recommend it. So then an illustration now for osmosis is having this selectively permeable membrane that only allows the cross of water but not glucose. And in this side A, you have very little glucose. And in side B, you have a lot of glucose. You don't remove this membrane, but then with time, side B is going to increase in volume because you will move water from where you have low concentration of glucose to where you have more concentration of glucose. So this one would shrink, while this one will increase in volume, as you can see here. So this is what happens again when we have our cells immersed in this fluid with high concentration of sodium or glucose. We dehydrate our cells. So that basically is related to osmolarity and tonicity. Osmolarity is the total concentration of solute particles in a solution. And typically it is written as moles per liter. So tonicity is the effect of a solution on a cell volume. And then we have three types of solution or three types of environments. So we have hypotonic, hypertonic, and isotonic. Isotonic basically is the same concentration of ions or solutes, as another solution. And in this case, if the solution it's our cells or cytosol, we have to have that specific concentration of solutes in the IV line, for instance, that is given to you. In that case, you will have a normotonic or isotonic condition. Now, if someone is dehydrated, then the person has hypertonic environment inside the cells. Eventually, after the water is withdrawn in comparison to the extracellular fluid, and you will add water in the IV line so that the person can recover. But of course, it's much better if the person can drink the water. But let's say that someone is unconscious. In this case, you have to give water through the IV line without the person drinking. And then in hypertonic, when you have a 5% glucose solution, that in that case, maybe the person has edema, the cells are swollen, and with that hypertonic solution that you put, the person will withdraw water from the cells. And hopefully it's not a person who has renal failure, because otherwise that person will end up more edematous. So what are the effects then of these different solutions on the cells? Hypertonic solution will have the water exiting from the cell, and the cell will be shrinking. This is called crenation. Isotonic, no net movement of water, Water will still move, but basically you don't have any losses or gains. So the cell volume will remain. And then in hypotonic, the water will enter into the cells and potentially will burst the cells. This is called lysis. So this is then what happens to the cells. Specifically, this is a red blood cell, an erythrocyte. It's a disc-shaped cell, and if you put this cell in a normotonic or isotonic solution, if you magnify this plasma membrane from this erythrocyte, you will see movement in and out of the cells, but of water, of course, but this will remain that same size. And then, if you have your erythrocyte and then you put it in a hypertonic solution, these spheres that you see here outside the erythrocyte represent solids. It can be glucose, it can be potassium, it can be sodium. So in this case, the cell will detect a hypertonic environment and the water will move in effort to dilute the extracellular environment. So in this case, you will lose this water from the cytoplasm and your cell will shrink or crenate. And then when you have a hypotonic solution, in this case you have less molecules in this solution and you have a lot of water. Well, the water will move from the extracellular fluid to the cytosol and your cell will start becoming bigger. And this will potentially burst the cell. If it's an erythrocyte, this is called hemolysis. Hemo for blood. Okay, now let's move to see the different transport of molecules, but now it's going to be either a solid or a liquid. So cells can ingest, sometimes some cells can ingest large vesicles, and then they can release these large vesicles. When the cells releases large vesicles, this is called exocytosis. And when they ingest these large vesicles, this will be called endocytosis. Endo for inside. So taking in a substance is called endocytosis. Releasing it will be exocytosis. Some cells can digest the large particles. This is called phagocytosis. Like we have cells in the blood that are called neutrophils that can endocytose bacteria, and then they can destroy the bacteria. So phago means eating. So cytos for cell. So it's a cell that eats. And then pinocytosis is a cell that basically ingests a liquid or a dissolved substance. And all of this requires energy and involves the membrane to extend towards these molecules that it's going to be ingesting, or encasing, in the case of exocytosis, encasing these molecules in a lipid membrane, and then that lipid membrane integrates into the plasma membrane so that you can release these molecules. So this is membrane remodeling. So here we have an example of phagocytosis of bacteria. So this is a phagocyte, a cell that can ingest this bacteria. So this is the vesicle that it is used by this bacteria, by this cell to ingest this bacteria. Everything starts when a bacteria lands in receptors in the cell. And then this bacteria is going to be endocytosed by this cell extending pseudophyte or pseudopods. So then you surround the bacteria, and then once it's internalized, this molecule, this vesicle, is going to be called a phagosome. And then you have lysosomes that were produced by the Golgi apparatus of this cell, and in these lysosomes you have lytic enzymes. These lytic enzymes are going to break down, hopefully, this bacteria, and then when this phagosome merges this lysosome, this merge is going to be called a phagolysosome. And then after this bacteria is destroyed, you will release the molecules outside the cells, and this is going to be called the exocytosis. For pinoendocytosis, so it's similar to what happened before, but in this case for pinocytosis, it is basically bringing molecules that are diluted in the extracellular fluid, and then you form a vesicle that has some proteins, and this you will call it the protein-coated pit, and then you endocytose or pinocytose this liquid, and then you will release these molecules, these proteins, and then you can make a lysosome that you can release through exocytosis. Now, you can have as well something that is called the receptor-mediated endocytosis, in which you will have perhaps a hormone that binds to these receptors of the hormone, and then once you bring this hormone into the cells, you will just grab the hormone, and then you will recycle the receptors back into the plasma membrane so that you can get more hormone. And then if there is something there that you don't need, of course you can discard. Exocytosis is the same as endocytosis, but in opposite direction. In this case, you will get rid of wastes. So, or maybe you are exocytosing proteins. So in this case, we have an electron micrograph showing you how these molecules has been released by exocytosis. So this is the vesicle that it's attaching into this plasma membrane. So the plasma membrane is not released, it's just only the product. Any questions about transport? Now let's look at the resting membrane potential, or RMP. So, excitable cells, neurons, and muscle cells, they work with electrical signals. And they have different concentration of ions, and with this different concentration of ions, they have a negative charge. That when the cell is not active, this negative charge is going to be called the resting membrane potential, or RMP. Now, the RMP is measured in volts and small units of volts, thousands of volts. This we will call it millivolts. So, typically, the resting membrane potential will be minus 70 millivolts. And this is how you write it down, lowercase m and capital letter B. So this resting membrane potential happens because you have high concentration of sodium out, while you have high concentration of potassium inside the cells. And then because you have a selectively permeable membrane, and you have channels that allows the exchange of ions. And you have a pump that maintains that concentration gradient of positive ions and negative ions in and out of the cells. So specifically, that pump is going to be the sodium-potassium ATPase pump that helps to maintain that concentration. So how does these ions, or this pump, helps to maintain the concentration gradient? So again, sodium is highly concentrated outside, while potassium is highly concentrated inside. That generates an electrical potential. And sodium-potassium pump is going to pump out three sodiums while it's going to pump in 2 potassiums, so that you maintain that concentration gradient. And you have to have ATP. Some anesthetics helps to reduce pain by blocking the action of this sodium-potassium pump. So you lose this concentration gradient, and then your cells, your nerve cells that detect pain, they don't send that signal of pain into the brain. And this will be local anesthetics that blocks this sodium-potassium pump. So 3 sodiums out, 2 potassium in. So you maintain more sodium out and then more potassium in. So this is the sodium-potassium pump. It has 3 gaps for sodium and 2 for potassium. So potassium will come in, sodium come out. And then you maintain high concentration of sodium out and low concentration in. Sodium is represented by these purple spheres, potassium by the blue spheres. So in this case, if you see at this pump, and if you see the outside of the cells, the extracellular fluid here in this upper panel, how many sodiums you have out and how many you have in? The spheres, the purple spheres represent the sodium. So how many you have out? Count together. 1. How many?
There's 9 and 2.
Yes. Almost this one out, right? So 7 and 2, and eventually this one is going to go out together with this, so you will make 10 and 1. And then how many potassiums do we have inside? 6 and 2 out. So that's more or less the ratio that you see there. And since this is an antiport, basically once you grab your 3 sodiums and you bring it in, at the same time you will grab your potassium and then you bring it out. It's almost simultaneous, and then you maintain high concentration of sodium out while you maintain very little in, and you have very little potassium out and a lot of potassium in. And then you have to have ATP. So ATP triggers the movement of this pump, and then you end up with inorganic phosphate. And you have to replenish back this ATP with a source of energy, which can be glucose, it can be lipids, and hopefully not proteins. Our body tends to save the proteins for what is important. But if you have glucose available, it will use it first, and then if you don't have it, it will start using the lipids so that you can convert it into glucose and then into ATP. So that's the resting member potential. Now let's look at the organelles, their functions, and examples of what can happen with these organelles if something goes wrong. So organelles are specialized structures in the cell that perform several functions. And one of these organelles will be the mitochondria. Mitochondria, it looks like a sausage. It is small, it has a double membrane. The inner membrane folds in and forms what we call the cristae. The inside of this mitochondria has a fluid that is like jelly-like, that is full with enzymes that can generate ATP. And in this area is where we actually generate the most ATP of our cells. So mitochondria has a matrix with these enzymes, and that's where we have the citric acid cycle. And we also have the formation of ATP through what we call oxidative phosphorylation in the electron transport chain. Function is to produce ATP through cellular respiration. We need to have oxygen to form ATP in the mitochondria. This is aerobic respiration. You can still form ATP without oxygen. This is called anaerobiosis or anaerobic respiration, but it's less efficient. And that's why we breathe. We need to have oxygen to generate energy. Mitochondria is a unique organelle because it has its own DNA and it has its own ribosome, so it makes its own proteins. And mitochondria is derived from our mother. So examples of dysfunction of this organelle. If you have mitochondrial disease, it comes inherited from mom, and you can have muscle weakness, neurological problems, and fatigue. And I was talking about inherited disease. Sometimes as we age, we lose number of mitochondria. That's why we become more fatigued. But also, there can be certain things that can damage the mitochondria, injuries. But anything that decreases your energy, in this case, when you have damage to the mitochondria, well, you expect to have some sort of problem in terms of generating energy. Now, the next organelle will be the ribosomes. So the ribosomes have an RNA and protein. And these ribosomes are made in the nucleolus, inside the nucleus. Now, the ribosomes can be subdivided into free if they're not attached into an organelle in the cytosol, or they can be attached to the rough endoplasmic reticulum. And the function is to make proteins by translating messenger RNA. So translation involves making messenger RNA in proteins. Your DNA will be transcribing into messenger RNA. It will make a long polymer of these ribonucleotides, and then you will make your proteins with that code. So what will be an example of this function? Well, you have a defect that prevents you from forming proteins, and that can contribute to some anemias like Diamond-Blackfan anemia, which is basically due to the lack of these ribosomes. And then endoplasmic reticulum is a network of sac of membranes or network of tubules that basically have ribosomes attached to it. And through these ribosomes, the messenger RNA can come in, and then you will make a protein. So function is synthesizing proteins, but also in the case of the one that is a smooth endoplasmic reticulum, you can synthesize lipids. Like the plasma membrane is made in the smooth endoplasmic reticulum, and also detoxifies the cells. Example of a dysfunction of the ER. Well, ER stress is linked to neurodegenerative diseases and diabetes. Now, something as well to keep in mind is that the cells, they can have different levels of organelles depending on the type of cell that you are considering. If it's a muscle cell, you expect to see more mitochondria. because that's a cell that is specialized for contracting and you need a lot of ATP. Golgi apparatus, it's a stack of cells or membranes as well, and inside this you modify proteins, and this stack of membranes is called the cisternae. So this Golgi apparatus can receive the proteins coming from the rough endoplasmic reticulum in a vesicle, and then it modifies these proteins, it gives them a tag, and these proteins goes into a specific area in the cell membrane, or they can be exported. So it's, again, some of you put in the lab report or told me, well, it's like the UPS. Yes, it can deliver these proteins. And also it can modify some lipids and deliver them. So examples of Golgi problems will be congenital disorders of glycosylation. So you don't add glucose into the proteins. Lysosomes are membrane-bound vesicles, round vesicles. They have digestive enzymes. Depending on the enzyme is how you would call your lysosome. Sometimes you have lysosomes that are filled with peroxidase enzyme. In this case will be a peroxisome. And it helps to break down waste with this enzyme and recycle cellular components. The organelles, when they get damaged, they can be recycled. So some of these lysosomes help to recycle these molecules from these organelles. And as well, when our cell dies, these lysosomes releases the enzymes, and then you have autophagy or autodigestion. Examples of diseases that involve the lysosomes will be Tay-Sachs disease that results from the lack of an enzyme that's called neuraminidase that can lead to toxic buildup of a molecule in the brain that can lead to neurological disorders. So organelles are defined as little organs of the cell. And you saw this before. So here is an animal cell with the plasma membrane with the double layer of lipids. Underneath you have the cytoskeleton. This cell is an epithelial cell, so it has a basement or a basal side, the apical side, and the lateral sides or surfaces. And then here we have the centrioles that can attach microtubules during cell division. This is the mitochondria, the open view. This is the closed view. Here we have peroxisomes, lysosomes, the nucleus, which is where you store the DNA, and then the nucleolus here, the chromatin, which is the name of the DNA, and then this is the rough endoplasmic reticulum. This is the smooth endoplasmic reticulum here, and then here we have the Golgi. And there's contents transport between the nucleus, the rough endoplasmic reticulum, and the Golgi. So then this nuclear membrane has pores through which molecules come in and out. Transcription happens here in the nucleus, translation in the cytoplasm. So what are the key steps for making proteins? So you have in your DNA genes that tells to the cell, okay, make this specific protein. And this protein will be encoded within this gene. Every 3 nucleotides form a codon, which is basically a signal that tells, well, you have to make this amino acid. And depending on the sequence will be how many amino acids of each kind of the 20 that we have and in what order. And then you form in the ribosomes basically the polypeptides. So this is translation. But you need to make messenger RNA out of your DNA. So this is transcription. Again, this happens in the nucleus. And then this transcript, this messenger RNA, exits the nuclear membrane through the pores, goes and then brings that information into the ribosome. And we have a type of RNA that is called tRNA, or transfer RNA, that will bring these amino acids attached to an anticodon, which basically has the complementary sequence to these codons from the messenger RNA, and then you make your proteins. And this is the genetic code, just to show you the example. So these are the first base, second base, and third base. Uracil, cytosine, adenine, guanine. Uracil, cytosine, adenine, guanine. And then in here, you will add this according to what you are making here. So your first and second base here, it is uracil, UU. And then this one will be for the first base of this third base. Sorry, for the first nucleotide of the third base, so this will be U. So 3 U's equals to put an amino acid that's called phenylalanine. And then if you add this C for cytosine, you will be coding for phenylalanine. And then if you change this for an A, coming from this third base, you will form leucine. If you have a G, you will also form leucine. So more than one type of triplet can form different amino acids. And then we have start codons like AUG, or forming a methionine. And then we have stop codons. Your proteins can stop elongating, so you have a UAA, UAG, or UGA. And out of these, you end up with all of these amino acids. So these are the names and the abbreviation. We will stop here and then we will continue on